Associated glands of alimentary canal. Liver. The liver is an organ present in vertebrates and some other animals. It plays a major role in metabolism and has a number of functions in the body, including glycogen storage, plasma protein synthesis, and detoxification. The adult human liver normally weighs 1.7 to 3 kg and it is a soft pinkish brown boomerang shaped organ. It is the second largest gland within the human body, located on the right side of the upper abdomen below the diaphragm. The liver lies on the right side of the stomach. Structure of liver, external structure of the liver. The liver is covered by two sheaths, an outer membrane, serous capsule which consists of visceral peritoneum and an inner glycans capsule of a thin layer of dense connective tissue. The liver is divided into two main lobes, right and left lobes, separated by the falciform ligaments. The right lobe liver is differentiated into right lobe proper and quadrate lobe and caudate lobe in inferior side. A pear-shaped sac-like structure called gallbladder is attached to the posterior surface of the liver on the right side. So to the posterior surface of the liver on the right side, gallbladder is attached. Okay. Internal structure of liver. Numerous thin branched septa-like tubercle extend inward from the glycans capsule, dividing each liver lobe into numerous minute hepatic lobules. Okay, means structural and functional units of the liver. An intralobular branch of hepatic vein called central vein form the axis of each lobule. Around the periphery of each lobule are 5 to 7 groups of tubes called portal triads. Each triad contains a branch of hepatic portal vein, hepatic artery and intralobular bile duct. The latter receives bile from the liver cells by the way of bile canaliculi. At the periphery of the lobule, the bile canaliculi empty into Herring's canals. About 80% of the hepatocytes or liver cells from the radial plates. Therefore, the radial plates of hepatocytes are separated by the hepatic sinusoid, that is vascular space sinusoids. Okay, the hepatic sinusoids are wide capillary like blood vessels lined by incomplete endothelium. Kaffir cells is equals to hepatic macrophages or stellate cells are also present in the liver lining the sinusoid, which are phagocytic that eat bacteria and foreign substances. Fat storing cells are also formed in liver. Ductile system and flow of bile. The bile produced in the liver is collected in bile caniculi which merge to form bile ducts. They eventually drain into the right left hepatic ducts arising from right and left lobes respectively, which in turn merge to form the common hepatic duct. The cystic duct from the gallbladder joins with the common hepatic duct to form the common bile duct. Bile can either drain directly into the duodenum via the common bile duct or the temporarily stored in the gallbladder via the cystic duct. The common bile duct and the pancreatic main duct joins to form the hepatopancreatic duct, which opens into the duodenum of the at the ampulla of the vata, guarded by sphincter of Oddi. The sphincter of Boyer is located at the outlet of the common bile duct. Function of the liver perform the following function: secretion of bile. Hepatic cells of liver secretes bile that play important role in the digestion of the fat. Deamination, it is the process by which the amino group is removed from the amino acids, resulting in the production of ammonia, which is converted into urea. Excretion. Liver synthesizes urea with the help of ammonia and carbon dioxide. Urea is panned out through the excretory system. Glycogenolysis. It is the conversion of glycogen into glucose by the liver cells with the help of glycogen secreted by the pancreas. Glycogenesis. It is the conversion of excess of glucose into glycogen by the liver cells with the help of insulin separated by the pancreas. Lipogenesis. It is the conversion of excess of glucose and amino acids into fats, which also takes place in the liver. Gluconeogenesis. It is the formation of glucose or glycogen from non-carbohydrate sources such as amino acids, fatty acids, glycerol, etc. Detoxification. Liver converts toxic substances into harmless substances. Example: harmful prussic acid formed during metabolism in all body cells is neutralized and rendered harmless by liver cells. Hemopoiesis. The process of formation of blood corpuscles is called hemopoiesis. Synthesis of blood proteins. The liver produces coagulatory factors such as factor 1, fibrinogen, factor 2, prothrombin, factor 5, 7, 9, 10, 11. It also synthesizes protein C, protein S, and antithrombin. Secretion of heparin. Liver secretes heparin, anticoagulant. Lymph formation. Liver is an important seat of lymph formation. Synthesis of vitamin A. Liver synthesizes vitamin A from beta carotene. Secretion of enzymes. Liver secretes certain enzymes which play important roles in the metabolism of protein, fats, carbohydrates in the body. Destruction of red blood corpuscles. The old worn out red blood corpuscles are broken down in the liver cells. Phagocytosis. The Kupffer cells of the liver engulf the disease causing microorganisms, dead cells, and foreign matter. Osmoregulation. The liver produces angiotensinogen, a protein which helps kidney in maintaining body fluid osmoregulation through the action of renin of angiotensin. Production of heat. Due to high metabolic activities of the liver, enough heat is generated which is essential for maintaining the optimum body temperature. Then storage. Liver stores glycogen, fats, vitamins like A, D, E, K, B12, bile in the gallbladder, blood, water, iron, copper, and potassium. The liver is the main organ for the breaking down of hormones after they have solved their messenger function to their target cells.